comment that you can give me that will help me give you more points would be really appreciated. So if you turn something in that that is all digital, like if you took a really clear phone picture of your osmosis in potatoes last night and turned it in, then you don't really need to turn in the hard copy, and you can just put a comment, chemist, clear picture submitted, or digital only, and I'll look at it. If something is late, but there's a reason for it, I was absent on Thursday, you told me I could do this, turn this in late, I get an extension on assignments, any of that stuff, super helpful. Because then when I'm scoring, I'll take those notes into consideration right when I'm putting grades in. Yeah.
If we haven't quite finished, like a detailed photo of it tonight.
You can. What works best for me is for you to just take a picture and turn it in now, and then turn in another picture later with a note. And what I'm going to look at, and here's why, because what I want to look at is how much progress have you put into it. I don't have to take off points for something being late. And if I look at something and it's really clear, okay, this person's just like working, they just need a little bit more time, then I'm good. If what you turn in is a piece of paper that has osmosis and potatoes at the top and that's it, then what it tells me is, well, you just didn't do it yet. And so turning stuff in right now is really important, even if you're going to turn in more later. And then let's see, what's next? The AP classroom work, and we'll talk more about this in just a second. Don't forget that you should be, I don't know exactly how you do it because I don't have your view of AP classroom, but you should be submitting your own, scoring your own work. Yeah?
Can I turn in pictures of this and then the questions on the secretary?
Yep. And then, then two more announcements. One, Unit 2 exam still on target for Wednesday, the 5th of November. We'll talk more about it next week. We only have class 2 times next week, Monday, Wednesday. Wednesday is not a full 90-minute period, it'll be an hour or so. So, and then you'll have a long weekend, then we'll have another Monday, and then we'll have the test. So I will post the study guide. In fact, I meant to do that today. I will, during class today, post the study guide for Unit 2 so you can start working on that, and then you'll have a nice long time with a couple of weekends and like some extra time next week to get that done. And then the last thing is, there's homework tonight. It is the 2.7 topic questions, which is one question, and it's a free response question. And here's what I want you to do. One, do the best you can. You might feel great about it, you might not feel so good about it, you might be in between, that's fine. Number 2, you can self-score your work. And I can't talk you through it because I have no idea what it looks like on your computer, but I know that you can do it. I set the option on for you to score your own work. You may have to play around. And if you need help, if you're trying to score it yourself and you just can't figure it out, we can do a Google Meet, and you can share your screen, and I'll tell you what to click on, because I know I can help you navigate it. AP Classroom can be a little clumsy sometimes, and so it's not necessarily obvious that like click here and click here and click here, and then you can score it yourself, but do it. And what you're gonna see is it's really tricky. The question is tricky, the scoring is even trickier. You're gonna look at your responses and be like, I don't know if I should give myself a point for this. That's part of the process. And then on Monday, we'll take some time to go over it. I will post, did I already do this? No. I have my response that I've written up, and I'll post that for you so you can see what I did, how I approached it. And then this will be the start of our FRQ practice, and we'll keep doing this, probably like not next week, but starting after that, we'll be doing about 1 FRQ a week. That's just going to be part of our routine, and you'll get better and better and better and better and better at it. Okay. Next, does anybody have any questions on the 2.4 to 2.6 topic questions? If you didn't do them, then they're gonna be late, and it's not a big deal, but I do want you to do them, it's really important. And if you, if you didn't do them, then you really have no idea what you missed. So take a look. I feel like this set is pretty good. Some of them are clunky, but some of them, like this one, is number 2 is an example. Number 2 is an example of one that I could see this on the exam, something like this. It's a model of how a channel protein influences the movement of a particle across a cell's plasma membrane. Here's the thing that's moving across the membrane. Here's the channel protein. Here's the plasma membrane. It's moving with the concentration gradient, so this would be facilitated diffusion. And then it says, investigator wants to know whether this membrane is involved in transport of a certain particle. What would you do to try to test it? So one thing you could do is add small non-polar molecules to the extracellular space and see if that, if the channel protein affects anything. Small non-polar molecules will diffuse across the cell membrane without a channel protein. This isn't going to help. Measure the rate of extracellular fluid into the intracellular space. Also, not going to help. That isn't going to tell us whether that particle is moving in. Add more of the proteins to the plasma membrane and measure the rate of particle movement. Makes sense. If you have none, you shouldn't see any movement. If you have one, you'll get some. If you have more, you might see a higher rate. And then remove ATP from the intracellular space and measure the rate. Well, if this were active transport, that might change something, but it's not. It's facilitated diffusion. This would not require ATP. So the correct answer is C, and that is a really, very much an AP style multiple choice question. So that is it. These are as I've told you before, but just as a reminder, number 1, my exams are going to become more and more and more like the AP biology exam until we get to about unit 4, at which point they're going to be pretty similar. Number 2, this work will help prepare you for the real exam in May, because the questions are going to look more like this than they did like the questions on our first exam or like the questions that you've gotten in previous classes. The people who write these exams are trolls, literal trolls. They're not human. They're like evil little grubby creatures that live under bridges. They don't like people your age, and they want you to do poorly on the exam. That's their life goal, is to make you look like you don't know what you're doing. So we need to beat the trolls. That's our class, our new class motto, which I'm establishing right now. Beat the trolls. Beat the trolls. That's what we're doing. Okay. All right. Any other questions or any questions on that? Okay. I told you that I was going to go through the most complicated piece of information that we're covering this section today. The work that we do next week is largely going to be self-guided. You're going to figure some stuff out on your own, and it is not complicated. It's relatively straightforward. The stuff that you did earlier with membranes, membrane permeability and cell transport, also not complicated, just a lot of vocabulary. Osmosis and related topics, this stuff often trips students up. So, why does it do this? Why does it sometimes appear over here? I don't want it to. I never want it to. I want it to be where I want it, but it doesn't stay. So the official title for this section is Tonicity and Osmoregulation, but really it's osmosis. That's what we're talking about. So tonicity, you've learned this before, almost certainly in previous biology classes. What tonicity does is it compares the concentration of 2 different fluids. So for those of you who don't remember, if you have a fluid, something dissolved in it, then the fluid itself is called the solvent. The stuff dissolved in it is called the solute. And the amount of solute per unit of solvent is called the concentration. In chemistry, and therefore biology, we typically measure concentration in molarity. How many moles of a substance there are per per liter of water. And a mole is 6.04 times 10 to the 23rd particles. So it's a number. So it's like a huge number of particles per liter of water. And they use moles because they're annoying, because of chemistry. Really the easiest thing would be to think about grams per liter, but we don't do that. We think about moles, and then anytime you mix up a solution, you first have to convert moles to grams using the molar mass, and then you have to weigh out that, and then you put it in, and then you mix it up, and then you have a solution that somehow, instead of just being like 300 grams per liter, which would make sense, you have 0.5 moles per liter, which doesn't. And either way, tonicity is looking at two different solutions. If they have the exact same molarity, then we call those two solutions isotonic. Now, isotonic is a comparative term. It's not that this one is isotonic. It's not that this one is isotonic. It's that these two things are isotonic to each other. When one is more concentrated than the other, we still use comparing words. Hypertonic solutions have more solute, more things dissolved in them, and less solvent, more of the dissolving stuff, than a hypotonic solution. It's still comparative. There is no such thing as a hypertonic solution, period. Like I have a teaspoon of coffee in here. It's got water, mostly it's water, and there's other stuff in there. There's the coffee, like the various oils and particles and stuff from the coffee, and there's some oat milk in there. So whatever was in the oat milk, oats and like carrageenan or whatever's in there, all mixed in. This has a certain concentration. Is this hypotonic or hypertonic? What do you think? Hypertonic, hypotonic. Anybody know? Don't answer, because there isn't an answer. You can't answer that question. We have to compare it to something else. This would be hypertonic to regular tap water. There's more stuff in it than there is in regular tap water. But it would be hypotonic to my can of soup that I don't have, because I'm out of soup. Can of soup has way more stuff in it, dissolved in it, than this coffee. So hypertonic and hypotonic compare two things. Where things get a little weird is that often we are comparing a hypothetical cell to a hypothetical environment, or a real cell to a hypothetical environment, or a hypothetical cell to a real environment, which means a lot of times our comparison is implicit. If I tell you that a cell is hypotonic, what I'm implying is that there's less stuff in the cell, less solute in the cell than in its external environment. Technically, there's no such thing as a hypotonic cell. It's hypotonic to something else, because those are comparison words. But we generally leave that comparison out when it's implied. Or if I say, we have a cell in a hypotonic environment, then what I'm saying is the environment has less solute in it than the cell. So there's this implicit comparison that happens a lot in biology. And it's unfortunate, because what it leads to is confusion, because people think, well, if I know the concentration of something, then I can say whether it's hypo or hyper or isotonic, and you can't. You need two concentrations. Two concentrations are either the same, or one is hyper and one is hypo. Those are your only options. You always have to have a comparison, okay? When we talk about osmosis, we're talking about water, and we're talking about water in environments that are not isotonic. So water is a solvent. It can be a solvent just like any other solvent. Alcohol can be a solvent, like, what's the nail polish? Acetone is a solvent. There's all sorts of solvents. But when we talk about osmosis, we're specifically talking about water being a solvent and there's stuff in it. Osmosis is a special kind of diffusion. Basically, what happens is sometimes water can move around, but other substances are trapped. So if we have, for example, a semi-permeable membrane splitting a container into two parts, that semi-permeable membrane may be designed in such a way that water can get across it, but other things can't. We have membranes that can do that. We have them at the school. They're called dialysis tubing. It's like really common. We use it for like treating patients who have kidney disorders, and we use it experimentally. We use it for filtering water. We use it for a whole bunch of reasons. This is not the same as a cell membrane. Cell membranes are semi-permeable, but in general, cell membranes aren't super permeable to water. Water can get through, but it's not easy. What makes water get through cell membranes are those aquaporins, those transport proteins that are built into the membrane of the cell. But the membrane itself is generally not permeable to water. So this can happen across plasma membranes or other things. Typically when we talk about osmosis, what we care about is cells, and what we talk about are other things. And the reason is because we can play around with other things experimentally more easily than cells. It's way easier to take dialysis tubing, fill it up with a solution, and put it into a beaker than it is to take a single cell and put it into an environment that is either hypo or hypertonic to the cell, and then see things move around. So experimentally, we always do this, but what we really care about in biology is what's happening in cells. Yeah.
What's the dialysis tube?
Dialysis tubing is a very thin, clear plastic membrane that they use in medical. They use for medical purposes. So dialysis is your kidney filters waste products from your blood. Basically, that's how urine gets made. And so dialysis tubing, they use it in hospitals for people whose kidneys aren't functioning correctly. So when, sorry, I didn't say this part yet. So when you have a situation where solutions are not isotonic, then what will happen is over a semi-permeable membrane that water can cross and other things can't, the water will move. And that's the opposite of what we would normally sort of anticipate. If you look at this diagram right here and ask yourself what's going to happen as things spread out, our intuition says, oh, well, these big red dots are going to spread out and they're going to be even in the beaker, which would happen if there weren't a membrane. But if you put a membrane here that these red dots can't pass, but the water can, something strange happens, which is the water moves in to the other side. It swells it up. And you can see this. It will actually change the relative level of the fluids such that the side where the water moves gets higher and the side where the water leaves gets lower. And there is an actual force associated with this. There is a pushing force in the direction that the water is moving. And this happens because of physics, because of the laws of thermodynamics. You don't need to understand the details of how, but it is inevitable. Yeah.
Does the water from the side with the red dots move over to?
The water is moving back and forth. But what happens is, because there is less water over here and more water over here, there's a net movement of water from where there's more of it to where there's less of it. Effectively, what's going on is the water is diffusing because the other stuff can't. Diffusion tends to even things out. And in this case, since the red things can't diffuse, but the water can, it will. And it tends to even things out. Right now, we have a real high concentration of solute and a real low concentration of solute. Over here, it's more even. Now, in this particular case, it will balance itself out after a while, because in addition to being pressure caused by the water moving this way, there's also pressure caused by gravity. And when those two forces equal, then the net movement of water will stop. But if we were to watch water molecules, they'll still be moving back and forth. Yeah.
Can the pressure cause one of the large molecules to move across a membrane?
Not necessarily. It depends on what the membrane is made of and how much integrity it has. For sure, things can break. So it's possible that you could set up something like this, and eventually there would be so much, pressure from something that the membrane would rupture, and then for sure it would go through. Okay, so what this means is there's movement of water toward areas where there is a higher concentration of solutes. We can think about this in three ways. One is water will move from where the solutes are less concentrated to where they're more concentrated. The second way we can think about it is water will move from where the water is more concentrated to where the water is less concentrated. There's less water per unit volume over here than there is over here. This is all water, this is only partly water. So the water is more concentrated on the left-hand side. It's actually diffusing from an area of higher to lower concentration. The way I remember it, water chases solutes. Water will always move from places where there are fewer solutes to where there are more solutes. While water will always move from lower to higher concentrations of solutes. Okay, and then this is osmosis, the diffusion of water across a semi-permeable membrane. And the details behind it, like the physics of it, are just not important, but there is force involved. It's actual force. There's actual pushing going on because of a lot of different physics stuff. Yeah.
Is osmosis normally caused by does the water move through the upper pores?
Yeah. So technically, when we are looking at the diffusion of water across a cell membrane, we don't really care whether it's happening across the phospholipid bilayer or through those aquaporins. If it's happening across the phospholipid bilayer, then it would be simple diffusion. If it happens through the aquaporins, then it's facilitated diffusion, and either way, it's diffusion of water. Yeah. Okay. All right. Other questions? All right. So the next thing to think about is something called osmoregulation. So if you start with this idea that water chases solutes, then what you can imagine is as cells move things in and out across their cell membrane, that should potentially affect where water goes, because the water chases the solutes. So if cells move particles outside or inside of their cell membrane, the water should follow. So imagine that you have a cell that, you have a cell that transports particles out of it. So here's an example of a cell where there's particles on the inside and there's some on the outside as well. If that cell moves some of its particles from inside to outside, then the inside concentration, the cell, will have a lower concentration of solute. The outside of the cell part, the extracellular area, will have a higher concentration of solutes, and water will move with it. So if a cell pumps ions or other substances out of it into the external environment, water will also flow out of the cell, largely through aquaporins, through osmosis. Yeah.
So is the hypertonic, hypotonic, and isotonic all regarding the extracellular space?
So the answer is you don't know. So in this case, right, in general, if you just use the term hypertonic, hypotonic, isotonic, you don't know what you're comparing it to. In this case, it says hypotonic solution. So what that's telling you is you have a cell in a solution that has less solute in it than the cell does. The solution is hypotonic to the cell. In this case, they have the same concentration. And in this case, the solution is hypertonic to the cell, it has more solute in it. Yeah,
So let's say that you say, hypotonic cell, it would be the last one, right?
if you say there is a hypotonic cell,
Uh-huh.
the implication is that you have a cell that has a lower concentration of stuff in it than its surrounding environment.
Okay,
Correct.
so it's just like
Yeah.
how you word it.
The problem is that it's a comparison, so you need two things. If you only give one thing, if I just say, this cell is hypotonic, the correct response to that is, to what? But a lot of times, you won't get that. A lot of times, if I just, if you look in a book and it's like, a hypotonic cell is blah, 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 and then you have to make an assumption. So in that case, my assumption would be, if somebody starts talking about a hypotonic cell, I'm gonna envision a cell that has some solute in it, in a solution that has more solute in it than the cell.
Okay.
That, we shouldn't have to do that, but we do because unfortunately people take shortcuts.
So they're like, if it's a hypertonic solution, like a hypertonic solution to something, because couldn't a hypertonic solution also be hypertonic in another solution?
Yes, brilliant. Yes, that's exactly right. So what she said was, couldn't a hypertonic solution also be hypotonic in a different situation? And that's completely right. If I have a solution here that is 0.5 molar, then that solution is hypertonic to a solution that's 0.3 molar. But it is hypotonic to a solution that's 1.0 molar. So we would never say, this solution is hypotonic, because it's not. It's hypotonic to this, but it's hypertonic to this. It's like taller and shorter, right? Like if Samson comes up here and stand next to me, I'm gonna look pretty short. And I guess that would happen if anybody was standing next to me, so I'm a bad example. But if you were to take a person of normal height and put them next to Samson, Samson will look tall. But then if we took an NBA basketball player and put them next to Samson, then Samson would look short. Samson isn't short, but compared to a basketball player in the NBA, he is.
They have to be at least 6'7".
Samson, you just made me feel short. I don't need this right now. Other questions?
You can take them out of the lease for it.
Order cases.
You offered.
So less complicated.
Okay, and then the opposite will happen. So if a cell is pulling ions or substances into it, glucose, sodium, whatever, then water will move along with it. Water chases these solutes around. As the cell moves them in and out, water flows in and out as well. And this can happen just like from happening. It can also happen when there's a physiological need for something to happen. So when plant cells, for example, they use water as a structural piece. They inflate themselves with water, they swell up, and the cell membrane pushes against the cell wall. And that's why plant leaves have like rigidity. And they will try to maintain that rigidity by, if the water levels in the cells start to drop and the leaves start to droop, then they will move solutes into the cell itself, which will pull water along with it, which will tend to cause the cell to get larger and inflated. Yeah.
Keep saying
It's not my fault.
it.
I felt like that was a reasonable answer. Okay, so this is osmoregulation. And then the last piece here, and we've got a couple of slides to talk about with this, is something called water potential. And so I've told you about some of the things that the AP trolls want you to know and be able to do that I think is a little silly. The statistics stuff. I think statistics is incredibly important. I think you being able to calculate standard deviation by hand is silly. What was the other thing that I thought was silly? I don't remember. This is one. This is one of those things. So here's the thing. Water potential is kind of a cool idea. The basic concept of it is pretty simple, which is that water will move around due to a combination of two things. One is osmosis, the other is physical pressure. If you have a cell and it's really, really, really concentrated with stuff in an environment that's not really, really concentrated with stuff, then water will flow in and that cell will swell. The osmotic pressure is inward, so water's flowing in, but the regular pressure, it's called hydrostatic pressure, is outward because the water is pushing out. And eventually it gets in balance. And we can figure out by looking at both of those things whether water will tend to flow in or out of a cell or any other thing. That's as much as you should need to know about water pressure or about water potential. Unfortunately, you need to know more. So this is what I just told you. There's actual forces involved. It's real pushing. It's not just like some theoretical thing. The first kind of pushing that we've been talking about has to do with osmotic pressure. That's what happens when water is pushing its way into something. It is literally a force. If we had a visible membrane in this particular situation, we would see that membrane curve inward because the water would be pushing it in as it moves. It's a literal force that you can feel and measure. But there's also another kind of force which has to do with just pressure. This can come from being in a container, like a cell or a balloon or a box, and the more stuff you put into it, the more pressure there is. It's like inflating a tire. Or it can come from gravity, and that has to do with the difference in height between two water columns. So if you have literally a situation like this, what we would see is that the liquid on the left would drop as water moves to the right. The liquid on the right would rise. That would mean there's a pressure differential there. There's more pressure here than there is here from gravity. And what happens is the water stops moving in a net direction when the osmotic pressure and the hydrostatic pressure are in balance. So the physical pressure, we can call it. They don't call it hydrostatic pressure in the equations we're using. They call it water pressure. Or they call it pressure potential is what they call it. Okay, so this is the important stuff. And it's cool to think about, and it's fun to watch, and it's observable and measurable. Yeah?
Is the hydrostatic pressure the gravity? Or?
In this case, it's caused by gravity.
Okay.
In this case. But in other cases, it might be caused by a container. So when you pump water into a cell, when cells move water into them, it literally causes them to get bigger. The membrane stretches. It's like a balloon. In plants, they take that balloon, which is the cell membrane, and they put it in a box, which is the cell wall, and then they pump it up so that the cell membrane is pushing against the box. And that's what allows the leaves of a plant to stick straight out. If I don't water these plants for a while, the leaves start to droop because their water pressure is too low. Yeah.
So essentially osmotic pressure is water's pressure, hydrostatic pressure is an external pressure.
No, not necessarily. They're both related to water pressure. So you'll see in just a second. Conceptually, what I think about here is physical pressure versus osmotic pressure. But it's coming from the water either way. The pressure here is caused by gravity pulling down on water. The pressure in a container, the water is experiencing the pressure. The container is pushing, but the water is pushing in the opposite direction. So, but it's not important that you can distinguish between that. Here's the stuff that you really do have to know. So first of all, this is the set of equations and constants and other information that you're going to be given on the AP exam. And I'll come back to this in just a second. Samson, your question, this is the vocabulary that we need to know. The overall movement of water is going to be determined by two things. One, the pressure potential. That's the physical part of it that doesn't have anything to do with osmosis. And the other is called the solute potential. And that combination of things is what we call water potential. So water potential tells us where water is going to go. And that is a combination of both the other kind of pressure, and osmotic pressure. And this is where I find this irritating, because I can tell you why they put this in here. They put all this stuff in here because they know that many students aren't good at math. Math is not easy for everybody. And so what they're hoping is that they can put some questions in the AP exam that you miss just because you don't feel like numbers are your friends. And I don't like that. That's not how I would prefer to do things. I don't think you are gaining anything particularly meaningful in terms of your understanding of real biology. But somebody over there, absolutely guarantee you that somebody at the College Board, or maybe like in one of the power states like Texas or California or Florida, this is their baby. Water potential is their baby, and they're not gonna let anybody take it out of the AP exam. So we all have to learn it. I'm gonna let the experts at Bozeman Science explain this to you, because it will be easier for you to see, and more importantly, the video is linked in this presentation, so you'll be able to watch it over and over again. Here's my piece. You're doing algebra. It's relatively simple algebra. What's gonna happen is you're gonna be given enough information to find an unknown. You may have to do it twice. So for example, this equation is gonna be on your equation sheet. You'll have some other information. You'll be given information in a problem, and they'll ask you, what is the temperature of the solution? Or what is the concentration of the solution? Or what is the solute potential of the solution? They may have you do it twice, because we actually have two equations. One that says the overall water potential is equal to the pressure potential plus the solute potential of a solution. And one that is specific to the solute potential. This we could just substitute right in here. Water potential equals pressure potential minus ICRT. It's a bunch of unknowns. And if you correctly identify the unknowns, if you correctly identify the variables that you're given, you can find any one of those things given all the rest.
What's the pressure potential?
The pressure potential is that hydrostatic pressure, the pushing force that you get from a container, or the pushing force that you get when you have two water columns of unequal length. It's a physical pushing. It's not caused by osmosis. Okay. Think about a balloon. If I blow air into the balloon, there's two pressures. One is pressure of my lungs forcing air into the balloon. The other is the pressure of the balloon itself pushing on the air that's already inside.
So it's like the pressure of the cell pushing on the water?
That is generally how we're going to be thinking about it. Yeah, the pressure of the container, which is usually going to be a cell. The problem is we can't say that universally, because a lot of times we're going to be talking about solutions in beakers. And the nice thing is, if it's an open container, then the pressure potential is 0. But if it's not an open container, then the pressure potential might not be 0. And again, what you'll see here is that understanding what all these things are is less important than being able to take a problem, identify known information, use that to find something that you don't know, and maybe use that to find something else that you don't know. So, it's the whole thing, it's not that complicated. What makes it ugly is the vocabulary and the Greek symbols. They use this psi for water potential and pressure potential and solute potential, and that's dumb, because it makes it look way more complicated than it really is. Like most of us, myself included, look at this, and I'm like, that's all Greek to me. Literally, those are Greek letters. That's why you think that. Yeah.
Sorry, could you re-explain the difference between the two?
Between what two? Pressure and solute potential?
Yeah.
Yeah. So this is the difference between pressure and solute potential. The solute potential tells us how hard osmosis is going to be pushing. So in this case, we have a bunch of solutes in here and none in here. There's going to be osmotic pressure in this direction. The solute pressure refers to that actual physical pushing force. Okay? There's also what we call pressure potential. That is caused by something else. In this case, in this drawing, that's caused by the fact that these two water columns are different heights. And so there's more weight here than there is here. There is literally water that is pushing down here because of gravity. And if we opened up the floodgates, it would even out, right? Like gravity tends to pull, like you don't, if you have a liquid without a barrier, then you won't have two levels in it. Everything will be at the same level. Yeah?
Is the pressure potential in that second section then, like, the difference between the amount of pressure in one side and the other?
Yeah, so in these equations, what you're gonna be doing is finding the water potential of one solution. Then when you compare that to another solution, you can figure out what direction water would flow. So you don't do both at the same time. You would calculate those two things separately. And they're not gonna give you a situation like this because their formula doesn't take it into account. Their formula doesn't actually take hydrostatic pressure, it only takes container pressure into account. And so, and that's why if you're given something in a beaker, it's either gonna be an open container and you can assume it's zero, or it'll be in a closed container and you'll either be told what the pressure potential is, or you'll be finding it. That's the thing, that might be your unknown. But you're never gonna have to deduce it. You're never gonna have to look at a situation and be like, oh, pressure potential equals something, something, something. You don't need to do that because this is all you're given. There's gonna be a number. You're either gonna be given that number or you'll find it. And if you're not given it, then you look through the problem carefully and you'll almost certainly see, oh, the pressure potential is zero because it's in an open container, and then you'll just go on from there. Like this is, the question that you just asked really gets at the heart of why this is silly. This is not, like we're only looking at a tiny little piece of it. Most people don't really understand what's going on here. And all they're doing is setting you up to miss some problems. That's all they want, is to like throw some things at you that you're gonna freak out about because it's a trident and a bunch of math. So we'll let the expert actually tell us what's going on. Isn't that like, I know that's rather subversive of me to talk about stuff like that, but I really believe it. Okay, I'm gonna play this video for you.